Good morning, my dear students. I am Dr. Venkat. Welcome to Aniruddha Tutorials. In this class, I am going to discuss about the human reproduction. The reproductive events are initiated at puberty. Reproductive events are initiated at the puberty. The reproductive events are as follows. One is the gametogenesis. Both male gametogenesis and female gametogenesis. After that, insemination. Insemination means the entry of spermatozoa into the female body. Into the female genital tract. This entry of the spermatozoa into the female genital tract is called as insemination. After that, fertilization. As a result of the fertilization, the zygote is formed. Zygote undergoes the cleavage. The first embryonic developmental stage is morula and then the blastocyst. The blastocyst comes to the uterine cavity, and that is attaching onto the uterine endometrium is called as implantation. After implantation, the gestation period. At the end of the gestation, childbirth occurs is called as parturition. So these are the reproductive events in the human beings. Gametogenesis. Okay, there is a little bit difference between the male gametogenesis and the female gametogenesis. The female gametogenesis is initiated at the embryonic developmental stage itself. But the male gametogenesis is initiated only at the puberty. Even the gametogenesis is initiated in the females in the embryonic development stage, but the reproductive events are occurring only at the puberty. Gametogenesis, copulation, insemination, fertilization, zygote formation, blastocyst, implantation, gestation and parturition. And now I want to discuss about the male reproductive system of the human beings. Male reproduction is as follows. The male reproductive system. Male reproductive system contain the following parts. The structures of the male reproductive system of human beings contain 1. A pair of testes, and the accessory ducts, accessory glands, and the male external genitalia. These are the parts of the male reproductive system. A pair of testes, accessory ducts, accessory glands, male external genitalia. And now I explain about all these structures one by one. A pair of testes. Testes are located inside the abdominal cavity before the birth. Before birth, testes are present inside the abdominal cavity. Testes are located inside the abdominal cavity. Testes are located inside the abdominal cavity. But at the age of 8th month, I mean before the birth, testes are located inside the abdominal cavity, but 8th month, the testes are descending into the scrotum. They descend into the scrotum. For that transport or the traveling of the testes into the scrotum, there are a pair of the canals. The canals are called as the inguinal canals. So through the inguinal canals, and these inguinal canals are connecting between the abdominal cavity with the scrotum. They convey the testes into the scrotum through the inguinal canals. So these are the connections between the abdominal cavity and then into the scrotum. In some children, the testes, they unable to descend into the scrotum. They retain in the abdominal cavity. If the testes retain or remain in the abdominal cavity, and that abnormality is called as cryptorchidism. That is called as cryptorchidism. I repeat again. In some children, the test is unable to descend into the scrotum. They remain as it is in the abdomen, is called as cryptorchidism. Generally, the length of testis is 4 to 5 centimeters, and the width of testis is 2 to 3 centimeters. When the testis enters the scrotum, there is a less temperature inside the scrotum. The temperature inside the scrotum is temperature inside the scrotum is at about 2 to 2.5 degrees centigrade less than abdominal cavity. Less than abdominal cavity. When the temperature is lesser inside the scrotum, less than the abdominal cavity, and that low temperature help in spermatogenesis. The low temperature help in spermatogenesis. So, the spermatogenesis occurs at the low temperature. Spermatogenesis occurs at the low temperature. Inside the abdominal cavity, there is more temperature. In the scrotum, comparatively inside the body, 2 to 2.5 degrees centigrade lesser, and that is beneficial and advantageous, and that condition is helping for the formation of the sperm is called as spermatogenesis. And this is the temperature, length and the width. When the testis descends into the scrotum, for example, this is the scrotum. Inside the scrotum, the testis is present. Scrotum is nothing but the skin, a bag of skin with hair. Inside the scrotum, testis which is oval shaped present, and the testis are attached to the scrotum with the help of the fibrous cords. These are the fibrous connective tissue cords. Fibrous cords are called as gubernaculum. Gubernaculum. With the help of the fibrous cord gubernaculum, testis attached to the scrotum, and the testis are conveyed to the abdomen with the help of a cord. For each testis there is a cord, and the cord called as the spermatic cord. This is the spermatic cord. Spermatic cord. For each testis there is a spermatic cord. Spermatic cord, again, this is present inside the inguinal canal. What is that structure? What are the structures which are forming in the spermatic cord? Spermatic cord is formed by one, connective tissue. Inside the spermatic cord, connective tissue is present, along with the blood vessels. Blood vessels are present, nerve, lymph vessels, along with another structure. Another structure is vasa deferens. Vasa deferens also traveling through the spermatic cord. So this is a spermatic cord that is attaching to the testis. It is formed by the connective tissue, blood vessels, nerve, lymph vessels and the vasa deferens. And coming to the layers around the testis. Testis is wrapped by, yes, this is the testis, oval shaped testis. And the testis is wrapped by 3 layers. There is inner layer, there is a middle layer and one more, the outermost layer. Innermost layer is called as tunica vasculosa. Tunica vasculosa. The name itself says tunica vasculosa means it is vascular. Vascular in nature. Vascular tunica vasculosa. And one more, there is a middle layer. Middle layer is called as tunica albuginea. Middle tunica albuginea. Middle layer called as the tunica albuginea. Actually, this is the layer during the embryonic development of testis. Tunica albuginea forms many number of septa inside the testis. For example, this is the tunica albuginea during the embryo development, and that forms the septa inside the testis. That forms the septa inside the testis. And these septa are forming the testicular lobules. Testicular lobules are formed by the tunica albuginea. I write here, the functioning of the tunica albuginea forms the testicular lobules. Testicular lobules are formed. For one testis, as many as 250 number of the testicular lobules are present. For one testis, as many as 250 number of the testicular lobules. Each lobule contain 1 to 3 number of seminiferous tubules. 1 to 3 number of seminiferous tubules are present. Seminiferous tubules for one testis, for one lobule. Each testicular lobule contain 1 to 3 number of the seminiferous tubules. I repeat again, middle layer is the tunica albuginea, and that tunica albuginea forms the testicular lobules. As many as 250 per one testis. In the human body, there are 2 number of the testes. Total number of the testicular lobules are 250. 250 is equals to 500 number of the testicular lobules are present in one male. And coming to each testicular lobule contain 1 to 3 number of the seminiferous tubules. Seminiferous tubules are nothing but the structural and functional units of the testis. Structural and functional units of testis. Units of testis are called as the seminiferous tubules. And coming to the outermost layer, and that outermost layer is called as the tunica vaginalis. Tunica vaginalis. Tunica vaginalis is a pouch of serous layer. It is a pouch of serous layer. It is a pouch of serous layer. And that tunica vaginalis is not a single layer. It is double layer. Okay, the double layer tunica vaginalis means there is a outer layer and also inner layer put together called as the tunica vaginalis. But I name the outer layer and also inner layer. Outer layer of the tunica vaginalis is obviously called as the somatic layer. Outer layer is somatic layer and inner layer is the visceral layer. Inner layer is the visceral layer. Between the somatic layer and visceral layer, the cavity is called as the coelom, is filled by the coelomic cavity, and that coelomic cavity works as a shock absorber for the testes. It is called as the coelomic cavity. Coelomic cavity inside the coelom. And this is all about the layers present around the testis, number of the testicular lobules. The lobules are formed by the tunica albuginea. For one testis, as many as 250 number of the lobules. Each lobule contains 1 to 3 number of seminiferous tubules. Seminiferous tubules are the structural and functional units of the testis. Okay, this is the testis sac and gubernaculum and the spermatocord, etc. Then the TS of the testis, I mean the anatomy of the testis is as follows. Structure of the testis. Structure of the testis. I mean the TS of the testis. In the transverse section of the testis, you can able to see many number of the seminiferous tubules. I told you that seminiferous tubules are the structural and functional units of the testis. It is as follows. Seminiferous tubule, it is one seminiferous tubule and one more. Yes. These are the number of seminiferous tubules called as the seminiferous tubules. Seminiferous tubules. Seminiferous tubule. Each seminiferous tubule is lined by two types of the cells. This is the wall of the seminiferous tubule. Is lined by two types of cells. One are these male germ cells. Seminiferous tubules are lined by two types of cells. One are the male germ cells. The male germ cells. Male germ cells can be called as the spermatogonial mother cells. They are also called as spermatogonial mother cells. Spermatogonial mother cells. Male germ cells are called as the spermatogonial mother cells. And these spermatogonial mother cells, they undergo meiotic division. They undergo meiotic division, meiosis. As a result of the meiosis, from these mother cells, spermatozoa are formed. They form the spermatozoa. They form the spermatozoa from the spermatogonial mother cells. I repeat again, each seminiferous tubule is lined by the male germ cells are called as the spermatogonial mother cells, and they undergo meiotic division, reduction division. As a result of the reduction division, spermatozoa are formed. And the other type of the cells also lining the seminiferous tubule, and these cells are called as the Sertoli cells. These are long cells, actually long big cells. These are Sertoli cells. The Sertoli cells They help in the nourishment. They help in the nourishment for the developing spermatozoa. For the developing spermatozoa, they nourish the spermatozoa Sertoli cells. Okay, these are the two types of the cells lining the seminiferous tubule. Okay, from these, the spermatogone mother cells ultimately, because of the meiotic division, the spermatozoa are formed, and these are the Sertoli cells. They help for the development, for the nourishment of the developing spermatozoa. Between the various seminiferous tubules, there is the connective tissue, and this tissue is called as the interstitial tissue. This is the interstitial tissue. Interstitial tissue. Interstitial tissue between the seminiferous tubules, and that tissue contains special type of the cells. There are blood vessels, arteries and veins are present, nerves are present, lymph vessels are present, connective tissue present. Along with this interstitial tissue also contain these cells are called as the Leydig cells or Leydig cells. These are Leydig cells or Leydig cells, can also called as the interstitial cells. Interstitial cells are Leydig cells. The purpose of the Leydig cells, they synthesize and release. Leydig cells, they synthesize and they release hormones, many number of the androgens. They release many number of androgens. You know, one of the famous androgen is the testosterone. They synthesize, release androgens, and one such kind of the androgen is the testosterone. So the source of the testosterone is the Leydig cell. Leydig cells synthesize, release the testosterone. So this is all about the seminiferous tubule of the human beings. I think you understood that. Thank you for listening to me.